Oh, this is okay, here we are again. Molecular biology, are you ready? No? Okay, so, a public service announcement before knocking on someone's office door, check their schedule and see if they're going to teach within the next 10 minutes.
Should somebody do that again?
Yeah.
Same person.
Yeah. So that's okay. I tell them I teach them shortly. But it's a nice habit to get into because then you don't like frustrate your faculty who are like last minute perfect. Okay. You guys heard this all week. Pick up exams if you haven't. Gene expression topics need to be finalized this week. Most of you guys are looking good. And keep remembering you need to be proposing something. You need to have a hypothesis generated from the data. Exam. Okay, gene expression. So we've been moving on to regulation of gene expression. And we've been talking about DNA elements and transcriptional regulators and promoters and enhancers and locus control regions, and a new one that I totally forgot to do, which I'm going to throw in today, called insulators. And then we started to talk about how transcriptional regulators bind to DNA for the different domains that are common to binding to the DNA. Or not domains, but motifs. Motifs, domains, depends on the size. Okay, so this is where we had ended. We were talking about zinc fingers, and in general, zinc binding protein binds zinc not to specifically do something with the zinc, but really just to stabilize the structure of the protein so the protein can do its job. And zinc binds to histidine and cysteine, and is helping to hold the structure of the protein together. Zinc is coordinated by cis and his, and the cis and histidine have properties specific to their amino acid side chains that allow them to bind to metals, and here zinc. Some gene regulatory proteins have multiple zinc fingers, or zinc finger repeats, and they'll make multiple contacts with the DNA. It will look like this, where each one of these will have a zinc finger. And see, the zinc here is really just holding this alpha helix to the beta sheet. So the zinc itself is not binding specifically to DNA. That's not its role. It's just holding the structure of a protein together in a conformation that makes the region that's going to interact with the DNA accessible. So actually, I went to the expo the other day and I saw someone's project, and I thought to myself, oh, we should talk about use of metal binding characteristics of histidine in protein purification. That was your poster. And I thought to myself, I'm sure you've connected those, but not everyone knows about protein purification methods. And it's kind of a cool thing to learn about, especially if you have any desire to ever do any research. And it also demonstrates the metal binding ability of certain amino acids, specifically here histidine. So this was known, and it's been used to come up with a way to purify proteins. So what you do is you actually, when you clone your gene of interest into your expression vector, you engineer it to contain a his tag, which has multiple hisses, usually 6 histidines in a row. And it's a his tag because whenever you tag a protein, you're tagging it to bind to something to purify it. And you can actually purify it, so you'll express it in E. coli or mammalian cells or insect cells or wherever, but often in E. coli. And then you've basically turned your E. coli into a protein-making machine, and it's full of this protein with the his tag. And you need to purify it so you can study it in some way. And you purify it by binding it to a nickel or cobalt containing resin. Why not zinc? It's just, I guess, not ideal for production in for this method. But it's using the metal binding properties of histidine to isolate the protein of interest. And this is how it looks. You'll have some resin, a bead made of agarose or some kind of solid material that's going to be bound to your the ion. In this case, it's cobalt, but frequently it's nickel. It also has the 2+ charge. And then you'll have your protein, which is a big scribble here, that has been engineered to express the his tag. And so it's called polyhistidine tag here. It could be any number of hisses depending on what you've engineered your protein to have. But in this case, they're just showing 2. And it's going to bind, because histidine binds to metals, like we've been talking about in the zinc finger. It's going to bind to your cobalt, and so then through that bind to your bead. And then what you can do is it'll stick to your column. So this is a column that you pour your lysate from your E. coli that's expressing your gene. You pour it over the top. It binds to your column. You wash it so all the stuff that's not your protein goes away, kind of like how an IP works. And then you elute it with something that binds more strongly than the histidine, and that elutes your protein of interest. And people do this anytime you're going to make a crystal structure. You have to overexpress tons and tons of your protein. If you're trying to characterize proteins that interact with each other, sometimes you'll do it this way. I just thought it was a cool thing to throw in, because you just think of, oh, the teacher's talking about a zinc binding domain. But it's actually been used, and some smart person who worked for a biotech company said, hey, we can use this as in a lab application to purify proteins. You need to purify proteins all the time. Well, it depends on what you study, I guess. If you're an ecologist, you probably don't need to. But if you're doing biochemical research or cell biology research or molecular biology research or any of that, you'll come across this. Okay? Any questions about this? I thought it'd be a cool thing to introduce. Do you guys understand the connection between me saying zinc finger and this, or are you wondering why I'm talking about it? Yes. You're wondering what I'm talking about. Because I'm trying to kind of illustrate the fact that some amino acids bind to zinc or bind to metal ions, and just their properties, the cysteine and histidine amino acids have properties that allow them to bind to metal ions. And people have actually used that characteristic to purify proteins by throwing a whole bunch of those amino acids onto the tail of a protein. You add a bunch of histidines, and then you incubate it with a metal ion. It's going to bind because it has that property. And then you can wash away what you don't want. Whatever's bound will stick, and that's how you purify your protein of interest. Okay? Good times. Okay. Let's go on. Okay, so in leucine, zipper, and helix loop helix proteins, so these are 2 more DNA binding domains. Coil. So coil looks exactly what it looks like. It's a helix and helix, so coils bind to the major groove on either side of the helix. So this is another way it can kind of look like it's sitting right on top there. The major groove of DNA can fit beta strands or connecting loops. So it's not just helices. I've shown you a lot of helices that kind of sit in the major groove. You can also sometimes have some beta strands that will sit in there, or some loops. Usually these kind of flexible loops will connect helices or beta sheaves, and sometimes those will stick in or form contacts with the DNA. So we talked briefly about this before. The bacterial MET repressor, it regulates the genes that encode the enzymes that catalyze methionine synthesis, right? And so there are these beta strands that will fit into the major groove. And the way it works out is methionine binding to the repressor changes the conformation of the DNA binding protein, and that is what allows it to bind to the DNA and repress. So lots of methionine, bind to repressor, change conformation, allows it to bind to the DNA and repress. Okay. So I'm going to take a moment because I realized I left this out and I wanted to look at the fruit again. Not really. It just, I thought it was funny to put this up here. Okay, so we were talking, this should have actually been when I was talking about all the DNA elements. I probably should have grouped them together. But because it's also a DNA element, but we're going to do it now. So the question is, what prevents gene regulatory proteins from activating the wrong genes? Right? You've got your gene here, and then you've got a regulatory element here. You might have another regulatory element here. It can be all over the place, right? What keeps it to activating the gene that it should activate versus running amok and activating other genes? Sometimes, because you can be thousands of base pairs upstream or downstream from your gene and still be an enhancer, right? So why isn't it, and sometimes there's, you're going to have another gene over here and another gene over here. Why isn't it activating that gene? How does it know to activate the gene that's right there? Has anyone ever thought about this? I actually didn't really think about it, and it was embarrassing to me when I finally did think about it.
Gene control regions identical in these two different places?
No, they're totally different.
So there's a difference in sequence. They're sequence specific, right?
Okay, so what I'm hearing is, are these DNA elements the same or different? Well, there's a difference in sequence. But how does that sequence know to activate the gene upstream or the gene downstream? Yeah.
Does it have to do with the folding and the purple molecule at the top?
Mediator?
The mediator, and what to do with activation of the mediator?
Well, a lot of these proteins that bind to enhancer regions do interact with mediator, but how do you know which way to go? Mediator is actually kind of universal to all genes, right? It can interact with different transcription factors and specific transcription factors and general transcription factors.
So it's the same mediator for all genes?
Same mediator complex. Maybe there's some differences, but generally, yeah. Yeah.
There's a whole bunch of regulatory proteins that have to be associated in order to get the transcription factor. So even if one is at the wrong part, could activate two genes, it's only going to activate one that has the whole protein. Is that okay?
Okay, so there's a whole bunch of transcription factors that need to activate gene expression, even if some are going the wrong way, if the other ones aren't there, it won't activate. That's one way to think about it. I mean, you're not completely wrong, but one major function of these transcription factors that bind to the enhancer regions is they recruit other transcription factors, the general transcription factors. So you can imagine if you get inappropriate recruitment of transcription factors, that would be bad. Yes?
Is the proper cascade effect from signaling?
Something about signaling? No? Okay, so everyone's really focusing, yes.
What was the issue with sequence specificity?
Well, there's no issue. But, right, yeah, shut it down. So the question was, well, if these control regions are sequence specific, wouldn't they just activate the gene specific gene? But I'm asking you why they're not activating another gene anyway. Yes?
Does it have to do with genetics?
Does it have to do with epigenetics? Epigenetics is involved, but at a slightly later step. Okay, I'm just trying to see who felt like reading all Delbers. Because that's the only way you would know this. Okay, so I'm going to give you a hint. It has to do with another DNA element that we haven't yet talked about.
What we mentioned earlier.
Insulators, right. So it has to do with insulator. And so insulators are DNA elements that are involved in regulatory domain compartmentalization. So compartmentalizing regulatory domains to the appropriate genes, right? You can think of it as a transcriptional unit. And so sometimes you'll have gene A and gene B here, and my quandary that I was trying to stir some discussion about was if you've got an enhancer here, what keeps it from activating this gene here? And what makes it just activate this gene here? Okay, that was the quandary. And the way it's solved is there are these DNA elements called insulator elements, and there are two types. Some are enhancer blocking insulators that will prevent an enhancer from acting on a gene. And another kind is a barrier insulator, which will prevent spread of heterochromatin. So you might have heterochromatin here, but you don't want it to spread. Remember, it spreads easily because these reader-writer complexes just recognize and modify, recognize and modify. Something needs to stop that from happening. So there are these insulator elements. And so whenever you think of DNA elements, you need to think what binds to it and what's happening, right? You've got DNA, it's not just the DNA itself that's preventing the enhancers from running amok. It actually says that in the textbook. I thought that was cool. It's the proteins that are binding to these insulator elements. So this is how it looks. This is right out of the textbook. This is a Drosophila polytene chromosome that is stained with propydium iodide. What is PI stain? You know, yes?
Oh, can you go back to the previous thing?
Yes.
I mean, so in this case, how can gene A be activated then? Do we have another enhancer with gene A?
So the question is, how will this become activated? Is there another enhancer? Yeah. So what it does is there are insulator regions between transcriptional units you can pick up. So you'll have an enhancer, some space, a promoter, your gene, your end of your gene, and maybe some other enhancer, who knows? And then you'll have an insulator.
Okay, so
And then
if
you'll have your next unit.
okay, so if we have insulators between enhancer and gene, so that gene won't be activated.
Right. Yeah, but if you have an insulator between an enhancer and a gene, it's going to prevent activation. If the insulator is active, which means if the insulator is binding to proteins that insulate.
So there's a regulation of insulators.
So there's regulation of insulators, yeah. Everything's regulated, right? Because that would be very dumb of our genome. If there were just these huge sequences that just shut everything off nearby, right? So it all depends on whether or not these other regulatory proteins, which are not well described, bind to the insulator. And I did a little reading because I was like, someone's going to ask me how this works. And in the textbook, it actually says it's not known. But I looked, and a lot is known, and it has something to do with forming structures in the DNA that prevent activity within certain regions. So you'll have two insulator regions that'll like loop out the DNA and kind of insulate a gene almost. But it's involved in many things. They even had cohesins involved. And I was like, whoa, this is getting too crazy. I have to go to class, and then someone knocked on my door. But anyways, but it's very cool actually. It's very cool. Because I never thought about it. What's the brain? I'm telling you, there's these DNA elements like thousands of base pairs away from a promoter that activate. Well, what's to prevent them from activating all sorts of things? And that's bad. When you activate things in an unregulated way, you get all sorts of trouble. Did you have a question? No. Okay. So this was PI staining the DNA, and then this is green staining this protein called BEAF, which is a Drosophila insulator protein, and you can see it tends to show up in these bands. And it doesn't say anything else in the book about it. And so I'll leave it at that. Insulator proteins bind to insulator elements and prevent either spreading of heterochromatin. Well, that's more of these barrier insulators, or prevent enhancers acting in the wrong direction. Right, so I was going to put another beautiful picture for you guys, but then I said, you know what, it's going to bring up too many questions. So we won't do that. Yes? You can assume that between each insulator is at least one transcriptional unit. That's a logical line of reasoning. You know what, so what's interesting is I was trying to study how one of the genes that I work on is regulated. So I was looking in the database of all this ChIP-seq data where they immunoprecipitate all these proteins that bind to DNA and look to see where they bind in the genome. And I found this one gene, CTCF, keeps coming up at the beginning of every gene, and I was like, that's weird, what is that? And I remember looking at it going, it's an insulator protein, and thinking to myself, okay, well that doesn't sound very specific, and moving on. And now I'm like, oh, it's an insulator protein that's found almost like frequently before every single gene when it's not being expressed, I guess. Yeah. Okay. How are we doing? Good times? Yes.
I'm going to talk about the regulation later.
No, we're not going to talk about it because it's a fairly, let me see. So this was done in 97, so it's not, or 95. So it's not that new of an area of research, but in terms of textbook, like things that are defined enough to go into a textbook, it's fairly new. When I looked it up on PubMed, there were tons of papers and reviews, so it's a very active area of research. But it sounds like, so I do know methylation. One thing that did come up, methylation can regulate whether or not these proteins will bind to insulators. So that's one thing that I saw for sure. But I'm not going to go through a whole mechanism because it appears to be not well described. Okay. Any other questions? Who's having fun? Good. I love it. Okay. Okay, so let's go on. Regulation of gene expression. So like if you had to reorder your notes, you would put that insulator stuff back with DNA elements. It's always good to categorize things, right? So we talked about DNA elements, category. We've talked about proteins that bind or types of domains in proteins that bind. It's always nice to look for themes when you're studying. It helps you to organize yourself. Okay. So we're going into the very exciting world of regulation of gene expression. I'm going to give you a bunch of examples. Just look for common themes and make sure you understand them. It can be easy to start. It's easy to start getting very confused because you'll be like, this binds this and this, this and this. Just kind of sit back and relax and look at it and try to look for themes. Okay, so in bacteria, genes involved in one cellular function are often organized into an operon. You guys have learned this before, right? And you'll have a promoter that basically binds RNA polymerase. And whether or not it binds RNA polymerase determines whether or not these different genes are expressed. And that will result in different RNAs and different proteins. And the example is the tryptophan operon, and that regulates expression of genes required for tryptophan biosynthesis. So it's many different genes under the control of one promoter. That's an operon. It's in bacteria. I did give you the example of the locus control region, but it's still quite different. Okay, so I don't think they're exactly the same, but there is an example of a way to regulate a group of genes in eukaryotic cells. Okay, so when tryptophan is abundant, it can bind and activate the repressor, blocking transcription. We already saw this, so I don't need to go too much into it, right? So you've got your promoter and your operator, and you have a repressor, and when a repressor binds to your promoter, it prevents RNA polymerase from binding, and you don't get gene expression, and it's all dependent on the conformation of your repressor. If your repressor is in the appropriate conformation to bind to that major group of your DNA in that specific area of the promoter, then it'll bind. So tryptophan, in this case, tryptophan is what binds to the repressor, changes the conformation, allows it to tightly bind to the promoter and prevent RNA polymerase. Whenever RNA polymerase is prevented from binding to a promoter, gene expressions should be off, right? So that's something, if you start to think of things in very broad terms, you can get bogged down with this operon, this repressor, this promoter, this. Forget about that for a minute. Just ask yourself, what's binding to the promoter, and is gene expression on or off? If RNA polymerase is being recruited, you can safely assume gene expression is on. If RNA polymerase is not being recruited, you can safely assume it's off, right? And usually a repressor or an activator is going to determine whether or not it'll be recruited. Okay, this is quite similar to the MET repressor. So if you're comparing, think about MET repressor and the TRP operon, and they're both involved in repressors binding to some kind of something that changes its conformation, so it can bind to DNA and repress. Questions? Thoughts? Stories? Good? Okay, so here's how it really looks. So binding of tryptophan to the repressor changes the conformation of the repressor. So here's your repressor, okay? And when tryptophan binds, it changes it. So see, there's this helix here and this helix here, and they're kind of tilted at an angle, and so they won't fit in the major groove very well. But when tryptophan binds, it straightens it out. It changes the conformation, so it can fit very nicely and bind tightly to the DNA and prevent RNA polymerase from binding. Okay? Yeah.
Is that attenuation, or is it something else?
Is that attenuation? Oh dear. Can you give me context for what you mean? Like, I mean, attenuation can mean a lot of things.
That's the thing. I've always been confused about.
Yeah, I mean, honestly, you can attenuate a virus. You can attenuate. Attenuate usually is like a way of saying you're inhibiting something, or yeah, kind of. So I guess it is inhibiting because you're turning off gene expression. Maybe come up with a context for attenuation. Like I always think of the polio vaccine, live attenuated virus. It's still alive, but it's not causing trouble. It's not going to cause disease. It can still replicate, but it has nothing to do with this. So I think that word is very specific to the context that it's used in, which might be why you get confused by it. Yeah. Okay. So, right, so the helix-turn-helix DNA binding motif in this repressor will only fit into the major groove of the operator if tryptophan is bound. So here's your helix-turn-helix. And you could go back when you're studying, you go back in your slides and look and say, okay, well, I've seen this TRP repressor before, and it came up when we were studying these different motifs, DNA binding motifs, and look at it and see how this helix-turn-helix motif fits into this and how it works, and that will help bring things together. Okay. Oh my God, we're going to be early today, I think. So repressors block transcription, and activators activate by promoting RNA polymerase activity. So this is a major concept. And so then this brings us to the concept of negative regulation and positive regulation, which people get very confused about for some reason. And I think it's because it's easy to get bogged down with little details, but positive regulation is turning things on, right? And negative regulation is turning things off. And if you stick with that basic understanding, hopefully you'll be good. So what does this show you? Okay, so in this case, you have negative regulation where a bound repressor prevents transcription. So a ligand binds and it actually removes a regulatory protein from DNA. So normally you'll have repression and some ligand will come and it will bind and then it will come off. Is that right? Yeah, so normally it's off, and then when you add the ligand, it'll remove the repression. And here you can have ligand binding that will actually, you know what? Ignore this. I'm remembering as I'm explaining this to you that this caused so much confusion last year because the way the book shows it. I'm going to find another figure that causes less confusion and replace it, and we'll go over it. Because I'm remembering I had to go over this like 10 times last year. Just keep in mind, negative regulation turn off, positive regulation turn on, and there are different ways that happens, and it all depends on whether you're going to have a repressor bound or not. Sometimes you can have a ligand binding to something that's going to allow something to bind to the DNA. Sometimes you're going to have a ligand binding to something that's going to remove something from the DNA. All sorts of things can happen, but just remember, if you have RNA polymerase being recruited because no repressor's there, something's being turned on. If you have RNA polymerase being inhibited because there's a repressor there, something's being turned off. And we'll keep it with that right now until I find a better way to show this. Okay? I feel like this is the end. That's the end. We are really early today. Do you guys want to talk about anything?
This is a good way of thinking about the positive and negative regulation by comparing the amino acid biosynthesis to lac operons. So it's like if you just compare like, does the cell need this thing around or does it not? And then like the cell's never going to make lactose. It is going to make tryptophan. So then you can just like order it following that as something like, I don't need to make lactose. When do you need to be able to break down lactose? When lactose is present. So then the binding of that ligand will induce the expression. Whereas if there's TRIP present, then you don't need to. And just following that logically is a good way of like keeping positive and negative. Like that's how I like it.
Yeah. We're gonna do the lac operon.
Yeah.
Maybe we can.
So just comparing these two
Yeah.
has always been helpful.
I find that just keeping it as straight as if it's going to be on or off, because things could be negatively and positively